Thanks, Clotilde. I really appreciate the invitation to be here, and I'm going to confess up front that Clotilde picked the title because I'm pretty sure that no one understands the microbiome, including me. But what I am going to tell you about is the efforts that we're making towards gaining that kind of understanding, and also some of the projects like American Gut that are helping us get towards that kind of predictive understanding of the microbiome, understanding how it affects our health. And I'll talk a little bit about how the technology that you've created has really been helping us in this effort to get a handle on this vast number of microbes that inhabits each of our bodies. So why is there so much excitement about the microbiome right now? It's almost like we discovered a missing organ that weighs about as much as our brain, but it has far more genes, far more connections, and possibly even more complexity. And so what I'm talking about, of course, is our gut, and specifically the microbes that it contains. I see some of you have chosen to bring lunch along, very brave. I promise not to critique your choices. But the microbes in the gut and also all over the rest of your body play all kinds of really important roles that we're only just beginning to understand now, in large part because of advances in sequencing technology. So there's a whole lot of things that microbes now explain. For example, they explain why you're not crazy when you go camping. Some people really are 10 times as attractive to mosquitoes as other people. And in large measure, the reason why is that different people have different skin bacteria that produce different volatile organic compounds that the mosquitoes home in on. Now, it turns out that the microbes we have a huge impact on our drug responses. So, for example, whether or not acetaminophen, the active ingredient in Tylenol, is toxic to your liver depends a lot on whether you have a particular group of sulfate-reducing bacteria. And then similar stories have been worked out for digoxin, which is a heart drug, cyclophosphamide, which is a cancer drug, and so on. So how you respond to medication can really depend on what microbes you have. And then finally, microbes are even linked to behavior. So if you're a fruit fly, at least, the microbes you have determine who you want to have sex with. We don't know if this is true in humans yet, but maybe it's just a matter of time before the right study is done, and we can find out. So how do our microbes do all this stuff? Well, in large part, it's because they outnumber us. And so you might be wondering, how much do they outnumber us? So there's huge error bars on all these figures, but one very commonly cited figure is that each of us consists of about 10 trillion human cells that make up our bodies. But our microbial component, primarily bacteria in the gut, makes up to 100 trillion microbial cells. So by that measure, you could say we're only about 10% human. You might be thinking, and especially here, you might be thinking, well, what about the DNA? Isn't it the DNA that really makes us human? So let's think about that for a moment. Each of us has about 20,000 human genes, depending on what exactly you want to count in terms of non-coding RNAs and so forth. But we carry around with us from 2 to 20 million microbial genes. So by that measure, we're maybe only 1% human. And just like we leave traces of our human DNA on everything we touch in ways that can be used for forensics, the same is true for our microbial DNA. So a few years ago, in work I did with Noah Freire at the University of Colorado at Boulder, we were able to show that we can match your hand up to the computer mouse you use with over 90% accuracy in many cases. So this came out in the journal PNAS a few years ago, but more importantly, it was on CSI Miami, so you really know it's true. Anyway, so in large measure, what my lab's been engaged in over the last decade is this project of figuring out how can we map our microbes, whether it's on our bodies, like I'll talk about today, or whether it's around the planet, as we're doing in the Earth Microbiome Project, which I won't talk about here, but which Illumina has consistently been a supporter of. And what we're doing is we're really taking a leaf out of the book of the explorers and biogeographers like Darwin and Wallace, who really mapped out in the 19th century where the large organisms lived around the planet. And so the intuition here is that as you go around the world, you see indicator species that immediately let you identify a scene as coming from one location or another, because those organisms only live in specific places. So what we're trying to do in the microbial world is very similar, although as those of you who have suffered through introductory microbiology know, they all pretty much look the same under a microscope, right? So instead what we do is we turn to DNA sequencing to identify them by traces of their genomes. So for example, in the human microbiome project, together with a group of about 400 researchers around the country, we collected about 4.5 terabases of sequence data, combination of shotgun metagenomics and 16S ribosomal RNA sequencing, to get both at the functions and to get at the taxonomic profile of the microbes that were there. And so the drawback to this, as is going to be very familiar to all of you, is that when you get the sequences back, it's very annoying trying to figure out who lives where in the environment from the data. So this, for example, is the first file of data from the Human Microbiome Project. You'll be able to see right away that it's 454 rather than Illumina data because of the read-length heterogeneity, right? But it's really difficult to figure out even that this is an oral sample, let alone anything about the microbes that are in it, looking at the data in this raw form. So the software that my lab has developed, including the unifrac distance metric for comparing different microbial communities, and the CHIME processing pipeline for taking data right from the sequencing instrument and converting it into visualizations that you can understand, essentially lets you take all of the HMP data and convert it from this sort of format into this sort of map that's a lot more understandable. Although you're probably thinking, hey, wait, I still don't understand what these little walls are. But I'll explain that in just a moment. One of the things we're trying to do right at the moment is integrate CHIME into base space to basically seamlessly provide this capability to anyone who's using base space to be able to get nice visualizations of their data sets. So what is this visualization? Well, what we're doing is we're taking all of the complexity in these different microbial communities and condensing it down into one point on this graph per microbial community. So all of the complexity in this oral biofilm, for example, is going to be just one of these green dots on this map. In technical terms, what it is it's a principal coordinate reduction of unifrac distances, measuring the distance between each pair of communities in terms of the phylogenetic tree, so the evolutionary distance between pairs of communities. So 2 points that are closer together represent more similar communities. You'll notice there's some big regions of color on this map, and so what those are the different sites of the body. So the mouth is very different from the skin, is very different from the vagina, and is very different from the feces in terms of the microbes that it contains. And so the algorithm that does this knows absolutely nothing about the samples. It knows nothing about the people involved. All it knows about is the similarities and differences between the microbes and the samples as read out by their DNA. And so this is a pattern that emerges automatically. As I mentioned, what matters is the distance between pairs of samples. And so if you take two samples from the same person in different sites of the body, they're very dissimilar from one another. So if I highlight one person's oral sample and their fecal sample at the same time point, you can see they're in totally different parts of this map. But then it's a little more difficult to relate those to what that difference actually means. So the great thing about microbes is that microbes are everywhere, and they're all evolutionarily related to one another. So we can use Darwin's insight that there is one tree of life that relates all organisms to take microbes from any community, whether it's a human body or different physical environments, and relate them all to one another on the same scale. So suppose we compare the mouth to a coral reef, right? You have these complex carbonate structures with all these biofilms where the microbes can hide. So if we say that the oral sample is like the microbes on this coral reef, then the fecal sample from the same person is as far away in terms of its microbes from that oral sample as the microbes in this reef are from the microbes on this prairie. So just think about that for a moment. What it means is that a few feet along the length of your body can make more of a difference to your microbial community than hundreds of miles across the Earth's surface. Okay? And now the fecal samples are all focused down in this region down the bottom, but that doesn't mean that they're all the same either. It just means they're more similar to each other than they are to the microbes at other sites of the body. So for example, if I highlight a second person's fecal microbial community, that's as far away from the first person's as the microbes in this prairie soil are from the microbes in the soil in this forest. So how far away is that, you're wondering? Well, remember in terms of your human DNA, you're 99.99% identical to the person sitting next to you. But in terms of your gut microbes, you might only be 10% the same. So there's a tremendous amount of heterogeneity in these microbial communities between different people. Okay, so this difference between people might lead you to wonder how stable the microbiome is. And so together with my partner Amanda, who's put up with a lot in the name of the microbiome, we assessed our microbes at 3 locations in the body, so the mouth, the skin, and the gut, every single day for a 6-month period. And so this is on a somewhat earlier version of the data, but the colors are the same with the mouth, the skin, and the feces. And essentially the dark points are going to be me. The light points are going to be Amanda. And this is one of the first things, this is what we did on our first sequencing run using the Illumina technology, actually, back on the GA2X, which was the first time we could collect tens of millions of sequences rather than a few 100,000 on the 454 platform we were using before. And so with that scale of data collection, we were able to collect these daily time points for 6 months. So this animation I'm going to show you is going to show you how the microbes in our skin, our mouth, and our gut change daily over the 6-month period. So I'll just start that going. What you can see immediately is how even within a person there's tremendous variation in the skin compared to the mouth, which is relatively static, and the gut, which is intermediate between the two. You can also see how the two of us retain our separate microbial identities during the 6-month period, which is pretty remarkable when you consider that we live together, we have all kinds of opportunities to exchange microbes with each other, right? And you can also imagine how useful this might be for tracking responses to drugs or to other therapies, for tracking disease progression. So I'll just spin that around, and so you can see that the traces at every site in the body have remained separate from one another, despite living together for the entire duration of the study. So this might lead you to wonder, where do our microbes come from in the first place? And if you have dogs or kids, as I do, you probably have some dark suspicions about that. All of which turn out to be true, by the way. So just like I can match you to your computer equipment by the microbes you share, I can also match you up to your dog. And Rob Dunn and Noah Ferrer actually found that the one factor that most affects the microbes on your pillowcase is whether or not you have a dog. And it doesn't matter whether you say the dog sleeps on your bed or not, it's still affecting your pillowcase microbes. But in all seriousness, what matters the most at the beginning is how we're born. So this is work we did with Maria Gloria Dominguez, now at NYU, looking at babies within 20 minutes after birth, and then their mothers within an hour before they gave birth. And so what you can see here, in red, what we have is all the vaginal samples from all the mothers. And then in pink, we have all of the microbial communities from all over the body of babies who were vaginally delivered. Whereas over here, in dark blue, we have all of the microbes from the skin of all of the mothers. And then in light blue, we have all the microbes from the babies delivered by C-section all over the body. And then the mother's oral microbes are very, very distinct, just like you'd expect from what I showed you before. And so this is just fascinating, and it may suggest some of the differences in health between C-section and vaginally delivered babies, where babies delivered by C-section have somewhat higher rates of allergies, of asthma, of atopic disease, and even of obesity. And all of these conditions have now been linked to the microbiome. So there's this possibility that the vaginal microbes are actually an adaptation that converge during pregnancy on the same state to coat the baby in the right kind of microbes to educate its immune system later on. So you're probably wondering what happened next. And this is some work we did with Ruth Lay's lab at Cornell, tracking the progression of one particular infant over 2.5 years of life, just looking at the fecal community. And so what we're looking at here is the meconium, the first fecal sample the baby produces. And so then the question is, and you can see it looks like it's in this vaginal region, which is what we would expect from the kid's delivery mode. And so then the question is, over 2.5 years, how much does it come to resemble the adult fecal state? How complete is the progression? Is it relatively smooth, or is there a lot of back and forth? What happens there? So each frame in this is going to be 1 week in the life of the kid's developing microbiome over these 2.5 years. And so what you can see is that sometimes there's tremendous change week to week, whereas other weeks it's relatively stable. You can also see that it's getting down towards the adult fecal state, but it's by no means a smooth journey. And it's fascinating to consider in all of this that sometimes these differences 1 week to the next, even in the gut microbiome of this one infant, are larger than the differences between any two healthy adults we saw in the HMP. So coming up here, it's fascinating. So the baby gets antibiotics for an ear infection. You see this massive regression followed by recovery in the microbiome. So that went by pretty fast, so I'll just rewind that and play it for you again. On administration of oral antibiotics for an ear infection, we see this tremendous regression in the microbiome, undoing months of normal development, followed by a relatively rapid recovery. And then by the time he gets to 2.5 years, you can see he looks more or less like the adult fecal state. And so it's just fascinating to consider what is the effect of these early life events. Is anything that you do just washed away in all this natural change that you have going on? Or do these early life events really matter for the later development of the microbiome? And so in this context, Marty Blaze's work, both experimentally in mice and epidemiologically looking across human populations, that shows that early life antibiotics have lasting effects on traits like obesity, is fascinating, and it makes you wonder what other sensitive or critical periods there are early in development where interventions are going to permanently change the microbiome and lead to an altered state that connects to health. We can also do this sort of thing cross-sectionally because the effect size in development is really large. So this is work we did with Jeff Gordon's lab at WashU, looking at kids ages 0 to 18 here, and then the average distance between each sample and the average of the adults in the same population. And so what you can see is that for an African population in red, South American in green, and the US population in blue, the trajectory of the approach to the adult state is very consistent, with most of the action happening in the first 3 years. However, just because you're approaching the adult state at the same rate with the same kinetics doesn't mean that you reach the same state. And so fascinatingly, what we found, and this is principal coordinates plots of the same data, we see that age is the major driving variable from the youngest kids in red to the oldest adults in blue. But where you wind up is totally different and depends on population. So the US microbiome configuration is totally different from what we see in the non-Western populations, which separate from one another in an axis that goes into the plane of the screen here. And so in this context, it's important to remember that although the HMP spent $173 million characterizing the healthy human microbiome, and Meta had spent about 50 million euros doing the same kind of thing in Europe, when you're only sampling healthy Western adults, you only see this tiny fraction of configurations of the microbiome that you might discover. You see totally different things when you look at kids, and you see totally different things when you look at non-Western populations. And then I should also mention that the hunter-gatherers that we've been studying in different contexts look completely different from these pastoralists as well. So there's a tremendous amount of human microbiome diversity out there still to discover. One thing that's particularly terrifying in this respect is the results of drug trials. So if you're only testing your drug, for example, in Africa or in China or in India, where it's a lot cheaper to do the trials, and then you're deploying it in the Western population, because the microbiomes are completely different between those populations, and we now know that your gut microbes are linked to the ability to process drugs, to detoxify them, sometimes even to respond to them, whether the conclusions you draw from one population will work when you deploy the drug in another population with a different microbiome, we just don't know at all at this point, although it's a very active area of research. However, to figure out the differences between these populations, rather than doing 3 populations that differ in genetics, in diet, and environmental exposure, we might need to do 600 populations rather than 600 people to really untangle those variables. So the need for much larger scale microbiome sequencing definitely exists. So you might be wondering, does all this diversity in the microbiome actually matter? Because I speculated about the effect of drug responses, but we don't have a lot of solid data on that. So the solid data we do have, and again this is accumulated over the last decade, primarily because it's now so much cheaper to sequence the microbiomes and find out who lives in particular populations, has now linked the microbiome to a whole lot of different conditions. So for example, the gut microbiome is linked to some conditions that you might have expected, like irritable bowel syndrome and inflammatory bowel disease. Some conditions that might be a bit more surprising, including colon cancer and atherosclerosis, and even rheumatoid arthritis in humans. The data is very solid. And then, for example, obesity. We've been working for about 10 years with Jeff Gordon on that topic, first in mice and then in humans. And so today, for example, I can tell you with 90% accuracy whether you're lean or obese based solely on your gut microbes. So on the one hand, this is kind of a cool trick. On the other hand, we don't think it has a lot of commercial potential as a test for obesity, right? Because I bet you can tell which of these people is obese knowing nothing about their microbes at all. But if we do that same classification task based on the human genome, if we take every SNP ever identified by GWAS and ask, can I classify someone as lean or obese based on their genome, we can only do that task with about 60% accuracy based on human genes versus 90% accuracy based on microbial genes. And there was a paper published in Nature just in January where they looked at over 100,000 people at the human genome level. And what they found is that taking every gene that they were able to link to obesity in that huge GWAS, they could only explain about 2% of the variance in BMI based on human genes. And fundamentally, things like the obesity epidemic cannot be a human genetic disorder because if you think about it, that increase in prevalence of obesity from 1985 to the present, either all of the skinny people would have had to have been sterile or all of the fat people would have had a lot of kids, right, to be able to change their allele frequencies. So it's much more likely that it's some kind of environmental or transmissible effect. And we can do the same sort of thing in mice where in addition to being able to do observational studies, we can do a lot more experimental studies. And so in mice, in addition to the conditions that I mentioned, obesity has also been linked to mouse models for what they're worth of things like multiple sclerosis and depression and even autism. Of course, the great thing about mice is the ability to do experimental manipulations to establish causality. So this is Jeff Gordon's gnotobiotic facility at WashU, where what you can do is you can grow mice in a bubble with no microbes of their own whatsoever. So they're both culture negative, and when you do PCR for the 16S gene, you don't find any band at all. So you can raise them without any microbes of their own in these germ-free bubbles, and then inoculate them with microbes that you think are important and see what happens. And we're setting up the same kind of facility at UCSD at the moment, so basically right under BRF2, the building where I work, we're currently in the process of scoping and installing this kind of operation. So what's great about this is that you can take, for example, in the case of obesity, these are obese mice, and these are wild-type mice from the same litter, where you can see that the obese mice are about 3 times the mass of the wild-type mouse. And so having established that there were microbial differences between the mice, the question was then, could you actually transmit the obesity by transmitting the microbiome? And so this was work in Jeff's lab where basically they took the fecal pellets from the obese mice that had a different microbial community, transplanted them into genetically normal mice with no microbes of their own, and then those mice became substantially fatter than the mice than their wild type, the mice that got the fecal transplant from their wild type litter mates. And so the fascinating thing was why they became obese. So with the OB-OB system, the reason why was improvements in energy harvest. So essentially the fat mouse is more efficient at getting nutrients from its diet. But that's not true in a second system, the TLR5 knockout system that we studied with Andrew Gewitz's group now at Georgia State. So in the TLR5 knockouts, there's the same amount of energy left behind in the fecal pellets. You can do bomb calorimetry on them and see that there's the same energy left behind. What it actually is it's a behavioral phenotype. So the genetic knockout and also the wild-type mouse that gets the fecal community from the genetic knockout gets fat because it eats too much. So it's a behavioral effect. You can cure them with antibiotics. And you can also cure them by just putting in their cage the amount of food a normal mouse would eat. So they're hungry all the time, but at least they're not obese. And more recently, we showed that you can also do this kind of thing across different species. So in another NIH-funded study with Jeff's lab, what we did is we took feces from obese and from lean twins, human twins, and transplanted them into mice. And again, the mice that got a fecal transplant from an obese human themselves became obese. The fascinating thing about this, though, is that we could actually design a microbial community based on the microbial communities from lean people that prevented them from gaining this weight. And so the potential to actually design a community that alters physiological properties is something we can now do in mice, although we can't do it in humans yet. And I should admit that we haven't yet designed a community that will slim down a mouse, let alone a human, although that's exactly what we're trying to do in the next phase of this project, which was just recently funded by NIH. And we can do the same kind of thing for malnutrition. So in a Gates-funded project with Jeff's lab looking at kwashiorkor, what we can do is we can take a fecal sample from a kid with kwashiorkor, a profound malnutritional deficiency that was originally thought to be due to protein deficiency, but the evidence for that is less clear now. And so anyway, if you take the fecal sample from a kwash kid, transplanted it into germ-free mice, those mice are basically screwed, so they lose 30% of their body mass within 3 weeks. They die if they're left untreated. but we can rescue them with the same RUTF, ready-to-use therapeutic food, a supplement that's used in the clinic for quash, where basically the kids that get the quash, so the mice that get the quash kids microbes respond dramatically to the RUTF and recover fully. The ones that get the healthy kids microbes don't have any particular ill effects and have a much smaller response to the therapeutic food, which is basically a peanut butter-based supplement. So what's really exciting about this is that the prospect that you could take sets of germ-free mice and then colonize them with fecal samples from one individual person, and then try out interventions in those mice, whether they're diet interventions or drug interventions or other kinds of therapy, and then figure out what is going to work for an individual human based on their microbiomes. Of course, the germ-free mice are hideously expensive, and we can't exactly deploy those to developing countries. But the idea is to use those to fit an in vitro model, and ultimately a primarily computational model, where from a small amount of sequencing, we might be able to predict what would happen either to the mice or ultimately to the person under a whole bunch of different therapeutic scenarios, and use that to guide therapy. Okay, so one thing you might be wondering is a lot of what I've shown you here has basically been about patterns in the microbial communities. And one thing you might want to know is how can you translate those to molecular mechanism. And this in large part is why I moved to UCSD, because the germ-free facility that's going in the basement right under my lab and right next to all the liquid handling robots that I have is also right next to all of Peter Durrestein's mass spec equipment. So we can really couple the DNA sequencing results to metabolomics and understand what's going on at a functional level. So the example I'm going to show you is collaborative work with Peter that came out in PNAS last week, so you're really getting this up to the minute. And what we're looking at, for reasons that will become apparent soon, is the skin rather than the gut. And what we're doing is basically looking at the skin because it's an interesting interface between us and our environments. And it's and it has a very rich collection of microbes on it. So what Peter and his wife Kathleen decided to do was basically study their own skin microbiology and metabolome. And so what they did is they collected 400 samples from around each of their bodies, and then Peter ran them through the mass spec, both with UPLC, QTOF, MSMS, and with MultiTOF to look at all the metabolites. And then we did 16S ribosomal RNA sequencing on the Illumina platform to look at the bacteria in each of those spots. They had actually planned to do 1,000 samples each, but by the time they'd done about 500, it was 8 in the evening, and it was clear that they were going to be up all night doing the other half of their bodies. So they bailed out at that point. But I think you'll agree that it's still quite a lot of samples and fairly interesting. So what you can see is from the mass spec data, there are some mass peaks that are distributed pretty much all over the body. There are some that are localized, say to the armpit or the belly button or the groin. And so we can start to build up this overall metabolite map of where are the metabolites over the body. And we can do exactly the same thing for the microbes. So for example, Staphylococcus, like you'd expect, is mostly in the moist areas like the feet and around the nose. Propionibacterium is mostly on the head and shoulders. Corynebacterium is more broadly distributed. And we can also start to figure out where do these molecules actually come from. And so this is a molecular networking technique that was developed by Peter together with Pavel Pevsner and Nino Bandera at UCSD, where basically what you can do is you can take a whole lot of reference standards and you can ask how do the spectra from those reference standards cluster with my unknown samples. And so what we're doing here is clustering the data that came from human swabs, from cultures of bacteria known to grow on the skin, from cultures of human skin cells in vitro, and then from beauty products and from chemical standards. And you can see that there's a whole lot of blue on this graph, and the reason why is that over 90% of the peaks that we identified were from beauty products, so shampoo, sunscreen, moisturizer, conditioner, and so forth. And so that's just fascinating, right? One thing we're looking at the moment is the skin microbes and skin chemistry of the Hadza hunter-gatherers, who are the last hunter-gatherers in East Africa. They still hunt and gather 95% of their calories, and they've never been exposed to any of those beauty products. So we should be able to get an idea of what those chemicals and what those microbes look like in the natural state, as opposed to when we're slathering things all over them all the time. So the other thing we can do is we can run additional standards where the chemical standards are available, and essentially look at whether the retention time in liquid chromatography is what we expect, and also look at what happens when you have various breakdown products that we conjecture are happening. And so, so the great thing about that is that we can use these maps to match up the microbes and the metabolites. So for example, I told you before that Propionibacterium was primarily on the face. So then we can take something like oleic acid, which as you can see is mainly on the face and the hands, and what you can see is that the oxidation product, oxidized oleic acid, is only present on the face where Propionibacterium is. So that gives us a pretty good clue that probably the Propionibacterium is catalyzing that particular transformation. So then what we can do is we can grab the Propionibacterium, grow it in culture, and then ask if we add triolein, which is the precursor that we think that it gets the oleic acid from, or if we add the oxidized oleic acid standard, do we get that only in the presence of the precursor? And so what we can do is we can basically prove that particular microbes are catalyzing particular reactions. So, so we're doing this initially in the skin just because it's so much easier to access and you can grow most of the microbes aerobically. But one of the things I have set up at the moment is a liquid handling robot and a plate reader in the koi chamber, so one of those big anaerobic bubbles, so that we can do exactly the same kind of thing, looking at biotransformations in the gut and other sites where the bacteria are harder to culture. Okay, so actually that's misplaced because I'm not going to return to that. Yeah, so you might be wondering, how you can place yourself on the microbial map. And I know that at the sales meeting, Clotilde handed out a bunch of these kits for American Gut, which basically lets you claim your own pin on this microbial map and find out where you are. So there's a couple of catches. One is that although we're IRB approved for this at Boulder, we're not yet IRB approved at UCSD, where all our equipment is now. So we're hoping that we can get that approval through in the next couple of weeks. But more importantly, only about 20 kits have come back out of the 100 that were handed out. So it would be really nice if the rest of those kits, if you have them, if you send them back in, you'll be able to place yourself in the context of the Human Microbiome Project data set that I'm showing you here, and all of the rest of the stuff from American Gut. So essentially this was something that Jeff Leach and I launched in 2012 over Thanksgiving, which of course is a time when a lot of people are thinking about their gut. And basically the idea was to exploit the decline in cost between the 454 platform, which we'd used in the Human Microbiome Project for the 16S sequencing, and the Illumina platform to make it available to anyone who was interested at a price point where most people could participate. Of course, it turns out that not everyone wants to know what's in there. These are middle schoolers learning that what we do in the lab is basically use robots and lasers to look at their poop. And although you can see that some of them are really intrigued and you know they really want to get into science for the robots and lasers, and who doesn't really? But a lot of people have been fascinated to find out. So this is Daniel McDonald, one of the grad students in my lab who's really been leading the effort, handing out kits at the Personal Genome Project meeting a couple of years ago, where we have a whole bunch of samples from people who are also having their human genome sequenced so that we can look at interactions between the human genome and the microbiome. And so at this point, the project's raised over $1 million, mostly in $99 increments from members of the public. We've sent out over 9,000 kits, and we've sequenced and released into EBI samples, about 4,500 samples at this point. So basically this project, like the Earth Microbiome Project, as soon as the data comes off the sequencer, we release it, and it's free to anyone for any purpose whatsoever, whether they're a researcher or a physician or an academic or someone at a company. The idea is to make the data available with no restrictions whatsoever on use. Of course, we do strip out the identifying information, like you probably don't want your credit card number linked to the metadata or that kind of thing. But all of the de-identified data is available, as is the full processing pipeline that takes you from the raw files in EBI all the way to all the graphics that we produce, both at the summary level on the whole population and what we send out to individual people. So the demographics are really interesting. So remember that NIH spent $173 million on the Human Microbiome Project, looking at 250 people. Whereas in American Gut, we have over 3,500 people at this point. So the discrepancy between that and the 4,500 samples is that there's a lot of people who have contributed more than one sample. And by the way, we don't necessarily just look at the gut. We're also cleared to look at the skin, the mouth, the vagina. We did have to send back the one semen sample that we got in the mail. because EH&S, the environmental health and safety people, were not happy about that. And there have also been some issues with reading the instructions. So the worst example with that, so the tubes have 2 swabs in them. And the reason for that is the idea is to get 2 samples so that if one of them fails extraction or sequencing, we can just extract the other sample. One of these tubes came back where one swab was brown and the other wasn't, and it was labeled 1 fecal plus 1 oral sample. So we just really hope they took the oral sample first, although we have no way of knowing what happened with that. But what you can see is we have a much broader range of ages, for example, than the HMP, which is essentially a study of 25-year-old medical students at WashU and Baylor. We also have a much wider range of BMI and other characteristics. So one thing that's very interesting is that when we compare the American gut to the American gut samples to the HMP, what we get is we get exactly the same overall pattern in the communities. And this is really remarkable because instead of physician-collected samples that are immediately frozen, we're looking at patient-collected samples that are just shipped in the mail. And in general, they've been sitting around for a few days at room temperature. But we did extensive controls before we started the project to show that you could still get useful information at the 16S level and probably at the shotgun metagenomic level, even from relatively degraded samples. And so this is really important for field applications where you just might not be able to get the samples back to the lab quickly. We also get pretty much the taxonomic distribution you'd expect with a lot of firmicutes and Bacteroidetes in the gut, and then small amounts of the minor taxa, again more or less minor taxa you would expect, including Fusobacteria and Actinobacteria, for instance. And so these are the actual results that you get at the personal level to help you integrate yourself into the population. You'll notice that this says unidentified. The reason for that is that the open source processing pipeline does not by default have access to the personal information that we need to retrieve the name or other details like that. So if you're running the pipeline pointed at EBI, it's just going to say unidentified. If you're getting the results back via the project, it'll have your name here. And by the way, we can also customize this for particular applications if that's useful. So given that these were handed out at the sales meeting, for example, you might finally be interested in the opportunity to find out exactly what kind of shit the salespeople were full of. Although, what would be really interesting would be to do it comparatively with, say, people at Iron Torrent and PacBio, which just produced some fascinating results. But essentially what you get is you get some information about what's in your sample. So you get your sample at the phylum level, like the firmicutes, Bacteroidetes, and so on, compared to the average of the whole population, and then people on the same diet, of the same gender, similar BMI or age. And then Michael Pollan, who was one of the first and one of the most enthusiastic participants in the project, and he has a particularly diverse microbiome, as you might expect if you've read any of his books about food, is also one of the reference standards there. And so then we have a table of the microbes where that you have most abundantly. Then we have a table of microbes where you have more of them relative to other people, even if they're not dominant in your particular sample. So we can infer that this person eats a lot of rhubarb or chard because they have a whole lot of oxalobacter, which is metabolizing the oxalate in there. And one fun party trick is to sort of tell reporters what they eat based on these tables, which is fairly effective. And then it also tells you a particular rare taxa that you have that most other people don't have. It also tells you if you're missing any of the common taxa that most people have but you lack. And then it shows you how your gut microbes compare to others. So you get your personal dot compared to the Human Microbiome Project at all of these different body sites. You get yourself inserted into the global gut population that I showed you before, so people of different ages and populations. We're integrating a bunch of hunter-gatherers into this at the moment, so the Matsis, some uncontacted Yanomami, and the Hadza, who I mentioned earlier. And then you can see how you compare to the rest of the American gut population, looking especially at the amount of firmicutes, which is one of the major phyla in the gut. At the scale of thousands of people, we're finding all kinds of associations we didn't expect. So you might have expected that your microbial diversity increases with your age, which it does. You might not have expected that it changes with the amount of sleep you get at night, or the amount of alcohol you drink, or all kinds of other factors. So So in terms of what affects your gut the most, for example, so what we're looking at here is the amount of diversity, and so this is the number of people you need to look at before you can find a difference of a particular size. And so you need about 5 people per group to tell the difference between, say, your gut and your skin, for example. One thing that's fascinating is that one thing that's fascinating is that things like inflammatory bowel disease and antibiotic use have very large effects on the diversity, in both cases decreasing it. But for example, we see a clear seasonal effect here. And then if you're looking at the community difference, again, body side is most important. Inflammatory bowel disease, again, has a huge impact on your microbiota, as you would expect from peer-reviewed published data looking at clinically defined populations. So it's great that we also find that in these patient-collected data. But one thing that has an effect as large as IBD is actually the number of different kinds of plants you eat. So basically, if you eat a few plants, 5 or fewer in the last week, you have a very low diversity community. Whereas if you eat a lot of plants, then you have a very high diversity community. And then you can see that we can even pick up things like sleep duration, alcohol use, and so on here. BMI has an intermediate effect. So you can see that we can pick up things that are much weaker than the BMI effect linked to obesity by having these large population sizes. And again, this speaks to the importance of having very large numbers of people enrolled in the study where we can read out their microbiomes. So I'll just end by showing you the most compelling case we have about why you should care where you are on this microbial map. And this is a case of Clostridium difficile-associated disease. So C. diff kills about 14,000 people in the US alone. It's one of the most common hospital-acquired infections. And when we look at the stool samples of people with C. diff, this is just to reorient you on this map. The stool samples of people with C. diff, shown by these stars, look nothing like the stool samples of healthy people. So you can see that they look almost more like skin or vaginal samples. So this is work we did with Alex Kreutz and Mike Sadowski at the University of Minnesota. Came out in Microbiome last week, and Yoshiki Vasquez and Antonio Gonzalez in my lab put together these animations like the other animations I showed you. So essentially what's going to happen here is that 4 of these people who you can see have a completely dysbiotic community are going to get a stool transplant from this one donor. And so again, much like the case with the infant that you saw developing, the question is how much progress is there towards the donor's stool community state in this healthy region down here? Is that progress smooth, or is there a lot of bouncing around? What happens during fecal transplant? So each frame of this is going to be one day in the life of these patients who have profound diarrhea that's resisted antibiotics for over 2 years, or they wouldn't be eligible for the trial. And they're going to the bathroom 20 times a day or more. What you can see is essentially immediately they move into the healthy state where the donor is, and then they stay there during the several months of follow-up of the study. And essentially all the symptoms are gone within a day or 2 after fecal transplant. And so the last major study comparing fecal transplant to antibiotic therapy for C. diff had to be stopped early, essentially because the fecal transplant was so effective, it was unethical to continue withholding it from the people who could benefit from it, who were on the antibiotic arm of the trial. So the question that faces us now is how many other diseases are like C. diff where we can identify a clear dysbiosis linked to the microbial community and then move you from a dysbiotic state into a healthy state whether by some microbiome-based therapy, whether it's fecal transplant, whether it's probiotics, whether it's prebiotics, whether it's targeted antibiotics or phage therapy or other drugs. And how can we tell you not just where you are on this map right now, but develop a predictive model of where you want to go and to tell you turn by turn how you're going to get there, developing a kind of microbial GPS. And our challenge really is to figure out how to develop this microbial GPS and make it so easy to use that, for example, even our children can use it. So you could imagine a kind of smart toilet that immediately processes your fecal sample, perhaps by sequencing or perhaps by something like mass spec or Raman spectroscopy or NMR, and delivers the results immediately to your smartphone and tells you, is your microbiome going in a good direction or a bad direction? And so, and then maybe gives you some advice on what you should do. So this may seem far-fetched, but if you think about your actual smartphone, I mean, this has about the processing power of a Cray 2, which was the fastest supercomputer on Earth in 1982, right? And if you talk to engineers on the Cray 2 team and you told them in a little over 30 years, the supercomputer that's like about that big and about that high, and it takes water cooling, a whole team of engineers to run it. It'll fit in the palm of your hand, and there'll be like 1 billion units shipped worldwide, and they're all going to be networked together. You can only imagine what race of geniuses they would have assumed that humanity had evolved into to use all that processing power, and how disappointed they would be to find out about Twitter and Instagram and what we actually use it for. But the point is that what separates us from the Cray-2, that advance in DNA sequencing, as you know better than anyone, has been packed into 10 years rather than 30 years. And so in the next 10 years, we need to be thinking about what's separating the HiSeq from the equivalent of this, where we need to be thinking not just about research applications and industrial applications, but about consumer applications and entertainment applications, right? What is the equivalent of Twitter for DNA sequencing and DNA synthesis, where even your grandparents are going to want to use it? Because that reality is going to be here very soon, and there's not a lot of thought going into, at least as far as I know. Maybe you're thinking about it and it's just secret, but thinking about how is your grandma going to use this stuff? Because I think that will happen before very long. So with that, I'd just like to thank the large number of collaborators who've worked on these specific projects, most of whom I mentioned during the talk, our various sources of funding supporting them. The large number of people currently or formerly in my lab who've worked on this or other topics, many of whom I mentioned during the talk. And finally, I'd like to thank Clotilde for her role, both at 454 and here, in bringing us the technology that's really helped us advance it. And I brought you a copy of my book, which is going on sale today. So this is Follow Your Gut, The Enormous Impact of Tiny Microbes. And so this is for you. And anyway, if you want way more detail about any of this stuff than you want to know, you'll notice that it's a very small book because microbes are very tiny, and it's very, very friendly and non-threatening. So finally, thank you all for coming along today, and I'd be delighted to answer any questions that you have. And if we don't get to your question, feel free to email me. Thanks again.